Hello, my name is Dr. Ifeyinwa Okafor, a lecturer in the Department of Hematology and Blood Transfusion Science, College of Medical Sciences, University of Calabar, Nigeria. I really want to use this opportunity to appreciate all of you. You have been very, very supportive. Your encouragement has been awesome. Thank you very much for being part of what we are doing in Hematology and Blood Transfusion Science lecture series. And if you have not subscribed yet, please feel free to hit the subscription button. Today, we are going to be discussing another important topic in hematology. Permit me to share my screen with you. Today, we are going to be discussing iron metabolism. Iron metabolism. And at the end of this lecture, it is expected that you should have a good understanding of why iron is a key element in the metabolism of all living organisms. You should be able to know the minimal daily requirements of iron and understand iron distribution in healthy young adults. Mention key proteins that are associated with iron metabolism. Iron, as we know, is an essential mineral required by every living cell. It's needed for DNA synthesis. It's needed for oxygen transport. It is needed for respiration, energy production, and other metabolic processes that take place in the system. Atomic weight of iron, 55.85. You know, iron, together with other elements, shares other transition metals, shares two important properties of particular importance in biology. Two properties that is of particular importance in biology. They have the ability to exist in more than one relatively stable oxidation state. And they have the ability to form many complexes. So iron has the ability to form relatively two oxidation states, and iron also has the ability to form complexes with many. It has the ability to form many complexes. Iron's ability to accept and donate electron allows it to shuttle between two oxidative states, the ferrous, which is iron 2, and the ferric, which is iron 3. Now, this ability of iron to form these relatively stable two oxidative states is actually what helps iron to be part of critical enzymatic reactions that take place in our system, like oxygen transport, electron transport, or even in cellular energy production. This particular property of iron to form two stable oxidative states, ferric ion and ferrous ion, and its ability to move from one state to the other within the same system helps it to do what? To form part of these critical enzymatic reactions that take place in our system. The total body iron content of a 70 kg man is about 4 grams. And the largest component is circulating hemoglobin. The largest component, largest compound that contains iron in our system is the circulating hemoglobin. About 1 unit of blood contains about 450 ml of whole blood. And this 450 ml of whole blood contains about 200 milligrams of iron. Iron is part of being very important, part of many living cells, or the processes or metabolic processes that go on in many living cells. Free iron is actually toxic because this free iron catalyzes the formation of free radical ions in the system. So you hardly see free iron in the system. At any point in time, iron is incorporated into one protein or the other as hemoglobin, as myoglobin, as part of enzymes, or in storage form as ferritin, hemosiderin, in transport form as transferrin. So in plasma, free iron is actually very, very small. In physiologic state, in normal physiologic state, only small amounts of iron exist unbound in plasma. Very, very minimal. Iron is a key element in the metabolism of all living organisms. This can never be overemphasized because even in plants, photosynthesis, which is a very important process in plants, and important enzymes, ferredoxin, is part of that process. Iron is part of this enzyme. DNA synthesis, a very important process in human, also have enzymes that have iron as part of their components. Ribonucleotide reductase, a very important enzyme in DNA synthesis, also has iron. Also, bacteria and other nucleated cells cannot proliferate when the amount of iron in their system is insufficient. And then we have hemoglobin. When we say that blood is life, it's actually because red cells help in, among other functions, they help in doing what? Transporting oxygen. And the key thing inside red blood cell that transport oxygen is hemoglobin. And hemoglobin is made up of heme and globin. And the important part where iron is incorporated, where oxygen is incorporated, is actually the heme part. And this heme part is made up of protoporphyrin ring and iron. Heme is the site of oxygen uptake in hemoglobin and myoglobin. Heme is a part of oxygen uptake in the hemoglobin and myoglobin. Heme is also the active site of peroxidases that protects cells from oxidative injuries by reducing peroxins to water. Iron is also a part of heme, which is the active site of electron transport in cytochromes and cytochrome oxygenases, which is an essential coenzyme in Krebs cycle. In the root nodules of legumes, hemoglobin catalyzes the fixation of atmospheric nitrogen by symbiotic bacteria. This particular reaction is an important natural means of fertilization of soil and for the synthesis of plant proteins. So iron is a very key element in many metabolic processes that take place in all living organisms, be it in plants, in lower organisms, in animals and also in humans. Iron homeostasis in healthy individuals are actually balanced by these four processes that I have on the slide here. The nutritional intake of iron, that is the amount of iron we take in our diet, versus the amount of iron that we lose in form of blood loss, shedding of dead cells, or sweating and all that. And then current demands, the physiologic situation of an individual, the current demand of iron in the individual, and versus the recycled or internal iron supply. All these four processes give rise to what we call the iron homeostasis. The interplay between these processes give a balanced iron supply in the system. The nutritional intake, the amount of iron that is taken and the amount that is absorbed, and then the amount of iron that you lose through blood loss and other means, because body does not really have physiologic means of losing iron, but it can have acute blood loss or menstruation, and the minimal amount being lost through sweat, okay? And then current demand, because, for example, pregnant women need more iron than non-pregnant women, and then infants need more iron than adults. Then we have to talk about recycled iron. We know that red blood cells have a lifespan of about 120 days. And when these red cells are destroyed at the end of 120 days, they are catabolized. You know, the red cells, when destroyed, that is the hemoglobin, the globin part, and then the heme part. The heme part is further broken down to protoporphyrin ring, which goes down to form the bilirubin. And then the iron is released and recycled. The iron is released and recycled. That is, the iron part of the heme is reused in the system. It's taken back into the membrane and reused there again. So the balance of iron metabolism in healthy individuals randomly, reflect these variables. The nutritional intakes, plus the absorption amount that is being absorbed, the iron loss, the current demand, recycled and internal iron supply. The iron in diet, the food that we eat contains two forms of iron. The food that we eat contains two forms of iron, the heme iron and the non-heme iron. The heme iron and the non-heme iron. The heme iron is primarily found in meat, poultry sources, and the fish. The heme iron is primarily found in meat, poultry sources, and fish, while the non-heme iron is found in cereals, legumes, fruits, and vegetables. When we consume these things, cereals, legumes, fruits, and vegetables, as part of our daily diet, we get, non-heme iron from them. When we consume meat, poultry products, and fish, we have iron in form of hemoglobin and myoglobin from these products, heme iron as part of our daily meal. The normal daily diet contains about 10 to 20 milligrams of iron, mostly in form of heme. And out of this 10 to 20 milligrams of iron, about 5 to 10% is actually what is absorbed by an individual. And about 20% of heme iron and only 1 to 2% of non-heme iron is absorbable from our diet. The total body iron content is normally about 50 milligrams per kg in males and 40 milligrams per kg in females. And total iron in human body averages, like I said before, 4 to 5 grams in humans. Now, there are many foods that are actually rich in iron. In our environment, we can reflect, we can attest of so many food where we can get, that can serve as source of iron in our diets. I can only mention some. You can fill in mention other ones that you know from your local diet. I have, like I said here, meat products like liver, heart, red meat, and then we have poultry products like egg yolk. We have the chicken, have the beef, we have the fish, we have the cereal aspect of it, that the non-heme iron, the cereals, the beans, the soya beans, the grains, cereals, we have vegetables, all these contain non-heme iron. Now, there are minimal amounts of iron that you need to absorb daily in order for you to meet up with your daily requirement. And for you to meet up your daily requirement, you must take certain amount of iron in your diet daily. For infants, they need to ingest at least 10 milligrams of iron in their diet as part of their diet for them to absorb 1 milligram. And children can take 10 milligrams in order to absorb 1 milligram. And young non-pregnant women need to take at least 20 milligrams in order to absorb 2 milligrams of iron in their diet so that they can meet up with their daily requirement. Pregnant women need a higher amount of iron, at least 30 milligrams of iron in their diet for them to be able to absorb at least 3 milligrams to meet up with their daily requirement to produce hemoglobin. Men and postmenopausal women need less. Like they need to take at least 5 milligrams of iron in their diet for them to take in to absorb 0.5 milligram every day to meet up with their daily requirement. So as from here, you can see that the children, infants, young non-pregnant women, and pregnant women actually need to consume higher amounts of iron in their daily meals in order for them to absorb the amount needed to produce hemoglobin and other things that need iron in their system. Now, iron is found in many compartments. Iron is found in many compartments. We have what we call functional iron. Functional iron is like iron found as part of hemoglobin, myoglobin, or enzymes that are involved in many metabolic processes that are taking place in our system. We have storage iron. These are iron found as part of proteins that are used as stored iron, like ferritin, hemosiderin. We also have transport iron, iron that is found as part of transferrin, that is the protein that transports iron in plasma. Now we have hemoglobin iron. The iron content of hemoglobin is about 200 milligrams. And hemoglobin, like we said earlier, is one of the largest compound that contains the largest amount of iron in our system. And it takes up about 200 milligrams of iron, and this is about 67% of total body iron. We have storage iron, which is ferritin and hemosiderin, containing about 1,000 milligrams of iron, and this is about 27% of total body iron. We have myoglobin, containing about 130 milligrams of iron, and this is about 3.5% of total body iron. We have labile pool iron, 80 milligrams, which is about 2.2% of total body iron. Other tissue ions like contain about 8 milligrams, which is about 0.2%. We have the important one, the transport ion, which is about taking up about 3 milligrams of iron, but taking up just 0.08% of total body iron. That is the transport iron. Let us have a closer look at what I just said. Hemoglobin. Hemoglobin, which is about 0.34% iron by weight, contains approximately 2 grams of body iron in men and 1.7 grams in women. 1 milliliter of packed erythrocyte, that is packed red cells, contain approximately 1 milligram of iron. Now, hemoglobin contains about, the iron content of hemoglobin takes up about 67% of the total body iron. So hemoglobin is a compound that contains the largest amount of iron in the system, circulating iron. Myoglobin contains about 3.5% of total body iron. And myoglobin is structurally similar to hemoglobin. It is a monomeric consisting of a hem group, like hemoglobin, and polypeptide chain, containing approximately 150 amino acid residues. Myoglobin is present in small amount in all skeletal and cardiac muscle cells, where it may serve as an oxygen reservoir to protect against cellular injury during period of oxygen deprivation. And this myoglobin contains about 130 milligrams of iron and takes up about 3.5% total body iron content. Then we have tissue iron. The tissue iron normally contains between 60 to 80 milligrams of iron. And it takes up about, this tissue iron includes the cytochrome and the iron-containing enzymes. And we have, although it's a very small compartment, it is an extremely vital one that is sensitive to iron deficiency. As you can see, that's about 2.2% of total body iron content. But it is very, very essential compartments of iron because it includes those iron that are part of cytochromes and part of our enzymes that are involved in many metabolic processes in the system. Then we have the storage iron. Another compartment is the storage iron. And iron is stored either as ferritin, which is water soluble, or hemosiderin, which is water insoluble. Iron is stored either as ferritin, which is water soluble, or as hemosiderin, which is water insoluble. Let us look at ferritin. Ferritin is a protein that is actually produced by the liver, and is predominantly found in the liver, the spleen. It can also be found in the plasma. And the primary function of ferritin is actually to store iron. It serves as reservoir for iron. The protein share, that is the apoferritin, that is ferritin that is not without iron, is composed of 24 similar or identical subunits arranged as 12 dimers and is approximate in the whole sphere. The apoferritin monomers are of two types. That is, you have two different types of ferritin subunits, the H subunit and the L subunit. The H subunit is encoded by the genes on chromosome 11, while the L subunits are encoded by the genes on chromosome 19. The L monomers, the L subunit of ferritin, have 15 hydrophilic residues that may bind iron, thereby promoting its retention. L monomers they have this capacity to bind iron and promote iron retention. While H monomers have fewer hydrophilic residues, but they contribute in binding iron and also releasing iron readily. H monomers have peroxidase activity, thereby enabling apoferritin to take up iron and also release iron quite rapidly. So you find H monomers, that is the H subunit of ferritin, in areas where iron is readily taken up and also released there readily, like in plasma. So ferritins that are found in the plasma are mostly made up of H monomers, while ferritin that are found in liver and the spleen are mostly made of what? L monomers. Apoferritin that is rich in H monomers takes up iron more readily, but retains it less avidly than those ferritin composed predominantly of L monomers. So ferritin that contains H monomers takes iron readily, but releases it more readily, while ferritin that's made up of L monomers retains iron more. And ferritin that are made up of L monomers are found mostly in the liver and the spleen. Much of this storage iron in liver and spleen is in ferritin containing mostly of L monomers. Okay? Ferritin is found in virtually all cells of the body and also in tissue fluids. In plasma, ferritin is present in minute concentrations, and it is glycosylated and largely composed of H subunits, like I said earlier. The plasma ferritin concentration usually correlates with the total body iron. And that is why measuring serum ferritin or plasma ferritin can help us to assess iron status of an individual. Remember, I said that ferritin is of two types. The H, it has two monomers, that is two subunits, the H monomers and the L monomers. The ferritin found in plasma and most body fluid are mostly composed of H monomers, while the ferritin found in the liver and the spleen mostly made up of L monomers. And this is because the H monomers has the ability to take in iron readily, but release it easily, while the L monomers takes in iron readily, but retains it more. For iron to be mobilized from ferritin into circulation or into transferrin, which is the transport ion, the ion needs to be converted back from iron 3 to iron 2, and also it has to be released from the apoferritin shell. As it passes from the cytosol to plasma, iron needs to be converted back again, reoxidized back again from iron 2 to iron 3. And the proteins that do this are what we call hepcidin, which is found in the duodenal enterocytes, and ceruloplasmin can also be found in plasma, and cyclopen found in the placenta. This enzyme that I mentioned, these proteins helps in converting iron from ferrous iron back to ferric iron for it to now be attached to transferrin for transport. Remember I said iron is stored in two forms, as ferritin and as hemosiderin. Ferritin is found in virtually all cells of the body, but predominantly in the liver, in the spleen. And the minute form of it is also found in the plasma. Ferritin has two monomers, the H monomer or H subunit and L subunit. The ferritin found mostly in plasma is in form of H subunit because it takes in iron and releases it more rapidly, unlike the ferritin found in the liver and the spleen that are mostly made up of L monomers, and they retain iron more. We have another form of storage iron, which is the hemosiderin, but the bulk of iron is stored more as ferritin in our system. Bulk of iron is stored more as ferritin in our system, and that is why ferritin can be used in assessing serum ferritin in an individual correlates well with the total body iron store of an individual. So if I want to assess iron status of an individual, one of the tests I can do to assess iron status of an individual is to do what we call serum ferritin, estimate serum ferritin. We also have hemosiderin, which is another iron transport, stored iron in the system. Unlike ferritin, hemosiderin is water insoluble. Is water insoluble. And most times, in order to assess it in the lab, we use what we call Prussian blue Pels reaction to assess hemosiderin. Hemosiderin is an amorphous structure with a higher iron protein ratio than ferritin, and it's probably formed by the partial digestion of ferritin aggregates by lysosomal enzymes. In normal subjects, the majority of storage iron is present as ferritin, and hemosiderin is predominantly found in macrophages rather than the hepatocytes. Hemosiderin is predominantly found in macrophages rather than the hepatocyte. Microscopically, in unstemmed tissues sections or marrow films, it appears as clumps of granules of golden refractory pigment, if you have overlooked at it under the microscope. Then hemosiderin, under pathologic conditions, iron may accumulate in large quantities in almost every tissue of the body in form of hemosiderin. You can see it being deposited in almost every tissue in pathologic conditions, especially in what we call hemochromatosis. You can see hemosiderin being deposited in every tissue of the body. Transport compartment. Iron is transported in form of transferrin. A protein called transferrin transports iron in plasma, while a protein called lactoferrin transports iron in milk. Like I said earlier, you hardly find free iron in the system. At any point in time, iron is incorporated into one protein or the other. Iron is incorporated into one protein or the other, either as stored iron in form of ferritin or hemosiderin, or as transport iron in form of transferrin and lactoferrin, transferrin in plasma or lactoferrin in milk. You also have iron as part of functional iron in hemoglobin, myoglobin, and as part of enzymes. So now we are discussing transport compartment, that is transport iron. The protein that helps in transporting iron in plasma is transferrin, while in milk you have lactoferrin. Now the transport compartment of plasma, is the smallest. It has about 3 milligrams, like what you said, and takes up to about 0.08% of total body iron stored. But it is the most active of iron compartment because it's always a turnover. Transferrin is always carrying iron to bone marrow. It's carrying iron to ferritin for storage. It's actually the one that receives iron from the duodenal enterocyte after absorption and carries iron to where it is needed. So those are normally turns over at least 10 times each day. It collects from ferritin and takes to where it is needed. It collects from the duodenal enterocyte through absorption and takes to ferritin for storage. It takes to bone marrow for erythropoiesis and also takes for incorporation into the formation of enzymes that are needed for Krebs cycle, DNA synthesis, and others. So this particular compartment, even though that is very small and takes up only about 0.08% of total body iron store, but it is very active compartment. It is very active compartment. There is the common pathway for interchange of iron between compartments. It is actually common pathway, interchange of ion between compartments. Transferrin and lactoferrin comprise a group of glycoproteins synthesized in the liver and by the cells of monocyte macrophage system. They are single polypeptide chains with molecular weight of approximately 80 kilodaltons. Transferrins and lactoferrins are synthesized in the liver and also by the cells of monocyte macrophage system. Each molecule has two binding sites for iron 3. Each molecule of transferrin has two binding sites for iron 3. Each is bilobed And within each lobe, the ion binding site is in a cleft between two domains that are designated as the amino terminal and the carboxyl terminal, N terminal and the C terminal. Within each lobe, ion 3 is bound to both the N and C terminal. So the key thing here is that ion is incorporated into transferrin as ion 3. So if ion is in form of ion 2, it must be converted to ion 3 before it can be incorporated into transferrin for transport. And the proteins that helps in this conversion is hepcidin in enterocytes, cellular plasmin in plasma, and cyclopen in placenta. They help in converting ion 2 to ion 3 so that they can be incorporated into transferrin for transport. Approximately 1/3 of the transferrin ion binding sites are occupied by ion, yielding serum ion levels that averages 120 microgram per dl in men and 100 microgram per dl in female. At least 30 genetically determined molecular variants of transferrin have been described in humans. What do we note? Key points here are: 1, iron is a very essential element, mineral, and it is needed by many living organisms and is incorporated as part of enzymes in many metabolic processes that go on in our system. Iron is part of hemoglobin. And hemoglobin, the heme part of hemoglobin, is the site of oxygen uptake in hemoglobin and also the myoglobin. Like I said earlier, so many enzymes in the system have iron as their component. Plants need iron as part of enzymes that are involved in photosynthesis. Many bacteria and cells cannot proliferate without iron. Even legumes need iron for the conversion of atmospheric nitrogen for the synthesis, natural synthesis of fertilizer. So you see, iron is needed by so many living organisms for important metabolic processes. And therefore, we cannot afford not to have iron in our diet. And we have iron in form of our diet, in form of heme iron and non-heme iron. And I said that heme iron, you can get it as when we eat meat, poultry products, fish. Non-heme iron you can get through cereals, legumes, vegetables, grains. When we consume them as part of our diet, we get it, we take in, and it's absorbed. Now, and also said that infants, children, pregnant women and young girls need more iron in our diet, in their diet, so that they'll be able to absorb the minimal daily requirement they need to produce what? Hemoglobin. While men and postmenopausal women need less iron. And we have key proteins that are involved in iron metabolism. We have ferritin and hemosiderin as storage iron. Those are the key proteins that store iron in the system. Iron mostly stored as ferritin, and ferritin has two subunits, the L monomer and the H monomer. The H monomer, taking in iron readily and releasing it readily, and it's mostly found in those places where iron is easily released, like in plasma. The ferritin found in plasma is made up of what? H monomer. While the ferritin found in liver and spleen is made up of what? L monomer. We have another protein that stores iron, which is hemosiderin. And hemosiderin stores iron, but mostly iron is stored in the form of ferritin. Then in pathologic condition, it can have this hemosiderin being deposited when you have excess iron in the system. It can have this hemosiderin being deposited in tissues of the body. Now, the transport compartment of iron, we have transferrin being the main protein that transports iron in the plasma. while we have lactoferrin being one that transports iron in the milk. Now, after taking in iron in our diet, the next thing we are going to look at is how this iron is being absorbed. When we take in iron in our diet, how is it absorbed in our system? So keep a date with us in our next lecture as we look at iron absorption, as we treat iron absorption. I know that you have actually learned something today. For further readings, feel free to look at these references. Feel free to look at these references for further readings. Thank you very much. Thank you, thank you, thank you, Anne.